My name is Erwin Skoof. I'm an associate professor here at the Technical University of Denmark, and I've been spending the last 5 or 6 years implementing single-cell proteomics workflows. Basically, we have this biological need to understand cell heterogeneity and how different cell types interact. In my case, both in a normal blood system, but also very much so in a cancer setting. So for this, we really need to be able to measure proteomes from single cells to figure out how different cell types are behaving, how they're responding to treatment, and how they give rise to biological phenotypes. So single-cell proteomics is really our workhorse. We combine it with other omics approaches like sequencing to really try and get to the bottom of this complex biological systems. For the last 20 years or so, we've been trying to do global proteomics, bulk proteomics, from which we get an average of a biological system. We can take a tumor, mash it up, read out what proteins were expressed, what was upregulated, what was downregulated, but it lacks this resolution of what's happening at the subcellular level. So what cell populations within a tumor are responding to treatment, which ones are not, how much tumor cell material is there immune cell infiltration, and all these things we basically miss out on by taking a bulk proteomics approach. And to this end, single cell will give us the more revolutionary insight. So single cell proteomics really aims to achieve quantitative accuracy, high proteome depth and high cell throughput, which are really the main challenges that we're trying to solve at this point. The thermo-scientific Orbitrap Astral MS spectrometer really presents us with a new horizon for single-cell proteomics. Not only because of the high scan speed, but also very much the added sensitivity compared to any other generation mass spectrometer that I've worked with. So the Orbitrap Astral MS spectrometer is really giving us a new level of data generation for single cells. So when we first tried our hands on it and ran the few first samples, we were expecting, better results than we've seen before, but suddenly we were getting both deeper proteomes and double the throughput in terms of single cells. So it was very clear right away that it was a revolutionary new mass analyzer that was going to lift the field to a whole new level. We really like having multiple mass analyzers in our instrument to play with. So in the case of the Orbitrap and the Astral, we rely on the Orbitrap for MS1 quant and the very high resolution and mass accuracy that this analyzer brings, combined with reading out the MS2s in the Astral, which is super sensitive and a very fast analyzer. And this is where the combination of the two really allows us to get the deepest proteome coverage while retaining accurate quant in single cells. If somebody had told me you can get an analyzer that has the sensitivity and the scan speed of an ion trap, but the resolution that's reminiscent of the Orbitrap, I would have jumped on it immediately. And actually what the Astral does is it actually did more than just that. It has very similar sensitivity as an ion trap, but even a much higher scan speed. So they've really taken this mass analyzer to a completely new level. For the first time I opened up a raw file from the Orbitrap Astral MS, I couldn't believe my eyes that we collected so many scans, more than 9,000, in just a 12-minute run. So just seeing the intensities of these MS2 scans coming from an actual single cell, I had never seen before. And then combined with the high resolution that we got from the Astral compared to what I was used to before from an ion trap was simply astonishing. It looked so much more clear and less noisy. I just knew this data search was going to be good. The emotion I felt was pure excitement, even a bit of disbelief. On the Astral, not only were we able to increase the number of cells per day to 80, so a double in throughput, we also increased the number of proteins per cell from 2,000 to 3,000 proteins. The deeper proteome coverage means we start picking up more key cellular decision-making proteins like transcription factors, epigenetic regulators, and kinases that we were actually not measuring very well before. So we get more of the important cellular decision-making proteins while also being able to increase our cell throughput. So now being able to run 80 cells per day in a label-free fashion, we start being able to run larger cohorts of cells in a relatively shorter time. meaning we can start unlocking the more complex biology where we need to measure a lot of cells in order to get to the bottom of the cell heterogeneity. But the research in my lab focuses a lot on making the world a healthier place, and I think that the Orbitrap Astral MS will really allow us to, in more detail, study how protein signaling is causing disease and causing specific treatment responses in, for example, leukemia patients, where we very clearly see that some patients respond to treatment, others do not and need to undergo additional therapy. And this is where the Orbitrap Astral MS will really allow us to measure these protein landscapes that are driving these biological phenotypes. So the throughput and sensitivity that the Astral Analyzer brings to single-cell proteomics will mean we can get much closer to the clinic. Single-cell proteomics could actually enter the clinic, since we have this throughput required to be able to make these real-time, real-life decisions. This is how we're going to have much more impact on human life.